Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
A common technique used to generate tumor cells for cancer research could produce misleading and unrepresentative results, a new study shows. Because cancer cells taken directly from patients are scarce, researchers often culture these human-derived cells inside successive generations of mice. They then study such, passaged, cells to see how they behave and how the disease might respond to treatment. The new findings highlight a serious limitation with this procedure. Human cancer cells passaged in this way end up very different from the cells in the original tumor samples. Transplantation into mice is the #1 preclinical model that people use. And that model itself induces some selection that people really hadn't appreciated, says Akil Merchant, a study co-author and physician scientist who works on leukemia at the Cedars-Sinai Medical Center in Los Angeles. Models are always limited. We know that, but now we have some insights into some of the very specific ways they are limited. The findings emerged from a study of cell heterogeneity in leukemia. We want to understand how these individual cells compete with each other and how they are different in terms of their growth, their metastasis, and also in terms of their response to chemotherapy treatment. Using a technique to read the gene expression of individual cells, and a DNA-based label. This is important because cancer researchers use mice to mimic the human body to study the disease and to analyze the effects of treatments such as chemotherapy. The study allowed the scientists to compare the cancer cells before and after they were transplanted into the mice, to follow cells as they spread around the animals' bodies, and to track how the cells expanded their populations and evolved through successive generations. Tracking the cell types when the mice received the chemotherapy showed distinct patterns of gene expression in cells that responded to the treatment. Different types of cancer cells tended to accumulate in different organs. To the surprise of the researchers, they also found that different types of cancer cells tended to accumulate in different organs. Cells with high expression of the CMC2 gene, which is involved in cancer metastasis, colonized the ovaries, while cells with low expression of CMC2 were found mostly in the blood and spleen. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.